Okay, welcome back to the last talk of the day. Before we get started, I will make the announcement that the plans for tonight's dinner have been solidified, and we will be meeting, or we will be leaving here at 5:30. But if you want to meet us there, it will be the restaurant or bar called Sin and Redemption. That sounds very exciting. At 6 p.m. And then without further delay, I'm very happy to be able to introduce Hildegard Uecker. I'm sorry, I know that's incorrect, but I'm sorry. Who is coming to us from Max Planck in another city I can't pronounce, Plön. And she'll be talking to us another talk about evolutionary rescue, general concepts and applications to multicopy plasmids and bacteria.
Yeah, so thank you very much for the introduction and for the invitation. It's really a pleasure to be here. So we've already heard about evolutionary rescue this morning, but just let me briefly recapitulate the scenario that I'm having in mind. So what you see here is the population size is a function of time, and the population is well adapted until the environment changes, and then the population size drops. And without evolution, without adaptation, the population would go extinct. However, it is possible that a well-adapted genotype appears and that is able to grow and rescue the population from extinction, which we call, as we've already heard this morning, evolutionary rescue. Such scenarios are of importance in conservation biology. That's kind of like what it also sounds like that when we want to actually promote persistence of species, but it's also of relevance in medicine, where like the evolution of drug resistance is an instance of undesired evolutionary rescue. And the same is true in agriculture when we think about the application of pesticides, where we do not want to have the evolution of resistance. So if you look at all this sort of resistance areas and themes, like in this very different fields, actually very similar questions appear. For example, that of which dose should we use? For example, here I have a paper, this is about fungicides, and this would be about antimicrobial chemotherapy. Another example is about drug cycling, which is looked at here in cancer, but also in the second one is about antibiotic resistance or herbicide rotations. And of course, like all of these systems have their specificities, but at a conceptual level, there are many similarities. And I think that these different fields can learn from each other. I'm having here a few more examples of what is kind of similar questions and similar topics across these various fields of resistance evolution. And especially if we are now thinking of the medical fields, there are of course even more overlap. For example, looking at this bacteriostatic or bactericidal drugs or the role of the immune systems and so on. And so my own research is mostly about, and it's an antibiotic resistance evolution. And I'm very interested to be here because I'm seeing in this talks a lot of themes appearing that are also important in the context of bacterial resistance to antibiotics. And so what I'm doing in this talk is I first give a bit of an introduction to the framework that I'm using. And there will be some overlap, I think, with Matt's talk from this morning. However, we coordinated a little bit. So I hope that it's mostly actually complementary to what he presented. And after that, in the second part, I will talk about some of the research in my group about antibiotic resistance evolution on multicopy plasmids. Okay. So if you're now going back to this evolutionary rescue, what does the outcome depend on, whether we have extinction or adaptation? And one is, of course, that this rescue genotype, it needs to be somehow present in the population, already present before environmental change, or appearing de novo during population decline. But then that's not enough, because usually it will only be there in low numbers, either maybe appearing just in one copy. And so there's a high probability of stochastic loss because even well-adapted individuals may fail to reproduce. Now, the second factor is a probability that this rescue genotype escapes from stochasticity. And both of these factors in turn can be influenced by genetic factors, by ecological factors, and often actually in antagonistic ways, which we will then also see during my talk. Let's look at a very simple model of resistance evolution here. And what I'm showing here is actually, it's like the continuous time version of this discrete time model from Orr and Ankles that Matt has introduced this morning. We are looking here at this sensitive cells. They have a death rate and a birth rate. The birth rate or cell division rate is smaller than the death rate. So this is an exponentially declining population. And the resistant cells, they can in principle grow because their division rate is larger than the death rate. We assume that maybe we are already having some resistant cells in the population when treatment starts. And they can also then appear during population decline because at cell division, cells may acquire a mutation. Okay. To analyze this, we look at this two factors. The first one, as I said, like the rescue type, or here this resistant cells need to be present in the population. And yeah, so there are already some present at the time of treatment onsets, but they can also appear during decline, which depends on the mutation probability, the replication rate of the sensitive wild type cells and the wild type sensitive wild type population, which we model here as deterministically decaying. But then also we need to look at this establishment probability. And as it's set up, it means this is now just a simple single type birth death process. Establishing is just the opposite of going extinct. We know what the extinction probability of this birth death process is. This is Q. It's just this death rate over birth rate. Now we put the two factors together, and that's pretty much the equation that you've already seen very similarly in Matt's talk this morning, where we have here this integral over the product of this mutational input and the establishment probability. That here, this is a zeroth term here, this exponential function of a Poisson distribution. That's a probability that no successful mutation appears during population decline. And the second factor is that none of the ones that have been present at the onset of treatment will establish in the population. So this would be our probability of evolution rescue, or like resistance evolution in our context. So now you may think, okay, this was such a simple model. Why do we analyze it in that way? Because I mean, you could also just model as a two-type branching process and then just get an exact result for the extinction probability or resistance probability of this process. And I want to show you why actually this approach has an advantage with a simple example. We are looking at bacteriostatic versus bactericidal drugs, asking under which one of them is a resistance evolution more likely. What I'm showing here is it has been done by my former PhD student, Christine. It's a tiny part of actually a huge paper on combination therapy. Now we are looking here at two types of drugs. There's this bacteriostatic drug. There's a replication rate, which is here just reduced by a factor. And there is this natural death rate of the bacteria. We are looking at the bactericidal drug, which the cells can replicate normally, but their death rate is increased by the drug. But that assumes sort of that the drug is killing independent of replication, which some drugs do, but many don't. And so we kind of take this into account in this third type of drug here, where we assume that the killing acts during a replication. So with a certain probability, a replication event fails, and instead of replicating, the cell dies. So this is added here to the death rate. We are distinguishing two cell types, sensitive and resistant. What you can see here is a net growth rate of the resistant type, of the sensitive type in blue. So that's the net growth rate as a function of the antibiotic concentration. You can also think, I mean, I'm thinking of antibiotics, but it could also be anti-cancer drugs, of course. And then we have the resistant cell, where we assume it has a cost. So its cell division rate, the cell division rate is lower in the absence of the drug than that of the wild type, but this curve is shifted to the right, so it can reproduce still at higher antibiotic concentrations than the sensitive cells. And for the sake of comparison, now we want to assume that in all of the 3 cases for this drug, the drug effect is actually the same for all of them for a sort of fair comparison. And so we can actually rewrite these terms for the cell division and death rates in terms of a drug effect. And you can see here for the bacteriostatic drug, it's just reducing the cell replication rate. For the bactericidal drug that kills independent of cell division, it's increasing the death rate. And in this last case, it's actually equally sort of distributed, reducing the replication rate or increasing the death rate. And you see that in all these 3 cases now, the net growth rate is the same for all 3 of the drug types. Okay. Now, this is now the probability of treatment success as a function of the drug concentration when we do not have any pre-existing resistance. And treatment success means we do not have rescue, so our population goes extinct. That's what we call the probability of treatment success. You can see here, I mean, if the drug concentration is too low, then we do not even kill the sensitive cells, so there is never any treatment success, but as we increase the drug concentration, it increases. And we can see that the probability of treatment success here is actually highest for the bacteriostatic drug. Now with the approach that I've introduced, we can also see why that is. Because we can look at those 2 factors. So what you can see here now is a total number of mutations that appear during population decline until the sensitive population is extinct. And you can see here the establishment probability of a single sort of resistant cell. And you can see here actually the bacteriostatic drug leads to the lowest number of new mutants, but the establishment probability is actually highest. And what we see is like if we put these 2 together, what is coming out of it is that this factor here, that there are fewer new mutations, is actually the dominant factor here. That's why the bacteriostatic drugs are better. However, what is also kind of already a little bit clear from that is if resistance pre-exists, then this one here has no sort of influence on how resistance evolution from the standing genetic variation. But we need to suppress very well the growth of this cells that pre-exist the resistance one. So we want to have a low establishment probability. And to have a low establishment probability, it's actually best to use these bactericidal drugs. And indeed, we see if we now look at the probability of resistance evolution or treatment, actually treatment success here, when we have pre-existing resistance, then the bactericidal drugs perform better than the bacteriostatic drug. Okay, so this approach that I've introduced with these 2 factors that can also be generalized to more complicated scenarios, but sort of the basic principle is the same. So you calculate how many pre-existing rescue types or resistant types there are in the population, and then how many are de novo generated during population decline. For this, one models the sensitive population deterministically because that's large, so that's a good approximation, and then uses branching process theory to calculate the establishment probability of the rescue or resistance genotype. And there can be, I mean, more complicated cases than the one that I showed here. Like it could be time inhomogeneous, for example, because drug pressure is changing over time or because of interactions with a sensitive population size, or we could also use multi-type branching processes and so on. And then we are putting it exactly together here again, building this product from the rate of appearance and the establishment probability, and here this term to manage the pre-existing resistance. Yeah.
Or even timing of homogeneous, it probably depends on parameters, but it's probability of extinction, and it is time dependent, right? Like, what if you only care if it doesn't go extinct in 5 years? You don't really take that key with you.
Yeah, it's not taken into account here. And like mostly we look at we just ask what is the probability, and we don't look at infinite times. But actually with the branching process, you cannot only look at does it go extinct ever, or like you can also calculate has it gone extinct by a certain time. So in principle, you can put it into the approach if you want. It's mostly not done, but I think it's a very, I mean, for many applications, it's actually very important.
Yeah, I can play around in the market. It's curious sometimes. Sometimes we don't care if it goes extinct. 100 years because no one will live that long. Right? So
yes. Yeah. Yeah. No, you're right. Yeah. Okay, so to sum this first part up, so this evolutionary rescue theory, it's kind of an umbrella for all the research that looks at adaptation to harsh environmental change that would drive a population extinct without evolution. And so the probability of evolutionary rescue can be calculated by looking at these two factors, the total mutational input and the establishment probability of rescue mutants. And I think what I also wanted to demonstrate is that this semi-deterministic approach, where we have the sensitive population deterministic and just the rescue mutant, which is rare, like stochastic, it has advantages because we can sometimes understand better what is going on, like maybe why one strategy is better than another. And also we can apply it to analyze more complicated scenarios where a full stochastic treatment would be difficult. Okay, so as I said, now in the sort of second part of the talk, I want to apply this framework to adaptation on multicopy plasmids. First of all, what are plasmids? So plasmids are extrachromosomal DNA elements, and they're very commonly found in bacteria. And so they have a specific copy number that depends on the plasmid type and on the bacterial strain. So for example, in this case, in this little example here, oops, it would be present in the cell in 3 copies. And just as a chromosome, they are also replicated and then transmitted vertically to the daughter cells. And sometimes can also be transferred horizontally to neighboring cells, but that's something we are not going to look at today. You should all be like care really deeply about plasmids because a lot of clinically relevant resistance is actually coded on plasmids, especially like resistance in bacteria that causes hospital-acquired infection. So what you can see here is a picture of Klebsiella pneumonia. That's a bacterium that causes hospital, like this ventilator-associated pneumonia in hospitals, and has often resistance on plasmids. Okay, so why do we care? As I said, I'm talking about multicopy plasmids here. So why do we care about the copy number of this plasmid? So the first is that the mutational input is higher if you're thinking about mutations on plasmids, simply because like if you're replicating the plasmid, there are more replication events, so more copies of the genes that can mutate. Second, when a mutation appears, that's what you can show here. So there's now this red plasmid. You can think of as a mutation, maybe a mutation then turning the bacterium antibiotic resistant has occurred on one of these copies, but it's then only present in one of the copies. However, when these plasmid copies are replicated and segregated to the daughter cells, we get then cells which have a plasmid composition that's generally different from that of the mother cells. And so over time, we are getting sort of this heterogeneous cell population where the cells differ in their composition with regard to these 2 plasmid types. And eventually, we are getting then also this homozygous types where all of these plasmid copies are mutated. So if now this mutation has a fitness effect, that also means that our cell population is heterogeneous in fitness. So what's shown here now is on the x-axis, that's a fraction of mutant plasmids. So here none is mutated and here all of them are mutated. And on the y-axis is the growth rate of the bacteria. And that here would be a dominant mutation because if just one of these copies in the cell carries a mutation, there's already the maximum fitness benefit a cell can have. But it could also be here the other extreme. So the mutation doesn't have an effect unless all of these copies in the cell carry the mutation. Okay, what we were now interested in was like, what is the probability of evolutionary rescue on multicopy plasmids? That was what my former student Mario studied. And for the sake of this talk here, I'm assuming that prior to this environmental change, for example, onset of antibiotic therapy, we do not have any pre-existing resistance. And then this population here, bacterial population decays, but mutations can occur generating this sort of rescue plasmids that may establish in the population and rescue it from extinction. Here's the model. So we have this cell here where this little n is the total number of plasmids in the cell. and I is a number of mutated copies. And so this genetic composition determines the cell division and the death rate of the bacteria. So it depends on I, and in principle, it can also depend on N. So what we are assuming here is now that the death rate is always 1 and the birth rate is 1 plus S, and depending on here's a fraction of mutant plasmids in a cell, call this sort of the dominance function, how this growth rate S depends on this fraction of mutant plasmids.
Is that during treatment?
That's now during treatment, yeah. So we assume that is during treatment, yeah. We assume that prior to treatment, like it's all just the sensitive plasmid. Then we still need to, that was just the cell dynamics, we also need to model the replication and segregation of these plasmid copies. So we assume here what we call regular replication. So prior to cell division, each plasmid copy is replicated exactly once. And then they are distributed to the 2 daughter cells in a way that, again, each daughter cell receives n plasmid copies, but with respect to the variance, it's random. So it's a hypergeometric distribution. Now, again, we need to exactly look at these 2 factors that I explained at the beginning, the mutational input, where we have this exponentially decaying wild-type population size. And what you need to keep in mind for later is here, now this mutational input is here proportional to n, little n, that's a plasmid copy number. And then we need to look at this establishment probability. Here now we are dealing with a multi-type branching process where a type is characterized by the number of mutated plasmid copies that a cell carries. So we can then calculate from this the extinction probability. So this Qi would be the extinction probability of a process where you're starting with a cell where I of the copies are mutated. And what we actually need for this establishment probability is the extinction probability when we are starting with a cell and where just one of the copies carries this mutation. And then we are putting this again together. As I said, I mean, there's no pre-existing mutations here. Again, what you have to keep in mind, it's important, it's this product of this mutational input and the establishment probability, and this mutational input is proportional to n, the plasmid copy number. Okay, so let's look first here at the establishment probability. So what is the probability that our mutation establishment probability doesn't go extinct if it appears? So I'm showing here results now for a dominant and for a recessive mutation. And you can see here now, it's this establishment probability as a function of the plasmid copy number, that in both cases, like both for the dominant and for the recessive mutation, it decreases with the plasmid copy number, which is also kind of intuitive because like if it appears in one cell and in one copy, and the more plasmid copies there are, the more cell divisions we need until we get this mutant homozygous cells, where then that's like mutant plasmid cannot be sort of lost in one of the daughter cells anymore at cell divisions. So and during all this process, it's always possible that a cell divides and one of the daughter cells doesn't carry this mutation, and it's kind of then just lost from this cell line. And that means like the more of the cell divisions we need, the more likely it gets lost. N is constant, yeah. Okay, so all of that was kind of now theory. And the question is like, do we also see this in experiments? And for this, we collaborated with a group at the University of Kiel. They are also interested in multicopy plasmids. And so the experiments have been performed by Anna, who was a PhD student at the time. And she really designed a super cool system that I'm now explaining to you. So what they are looking at is a plasmid that has a copy number of 15. And there's this sort of blue ancestral plasmid. And this carries a gene. There is this NPT2 gene. NPT2 that is a gene that gives resistance to the antibiotic kanamycin. But in this ancestral plasmid, it's broken. So they introduced here this GFP gene, so it's green fluorescent protein gene. So that means like an appropriate light, it's green fluorescent, but it means that the antiprototic resistance gene is broken, right? So these cells are not resistant to kanamycin. And then through a process that's called transformation, they are generating this novel plasmid where they have this functional NPD2 gene because this GFP gene has been recombined out. And so this transformation is basically that the bacteria are picking up DNA from the environment. And how this works, there's this it's called the one-hit kinetics. It's this novel allele with this functional resistance gene, then only present initially in one of these copies. So for comparison, they are also looking at a chromosome, which is such that a copy number of 1, where the ancestral allele is a TET-A, that it carries a TET-A gene that's giving resistance to tetracycline, so a different antibiotic. And the novel one is again this NPD2 genes giving resistance to kanamycin. And then they sort of let this population evolve over time in the presence of kanamycin. And what happens is exactly this process of plasmid replication and segregation that I've described. So they are getting then the cell population where sort of most of the cells, so this new plasmid was only introduced in a very small fraction of cells. So most cells are then now our wild type cells, but the ones that received this new allele on the plasmid, they are kind of doing this process of replication and segregation, then generating cells where more of these copies are actually having this novel allele. And finally, that's also generating then these cells where all of these copies of this plasmid carry the new allele. And they kind of can distinguish now these 3 types. So this ancestral type, where cells are homozygous for that, it's green fluorescent, but it's not resistant to kanamycin because this kanamycin gene is broken. The ones that are homozygous for this mutation, they're not green because this GFP gene has been lost, but they are resistant to kanamycin. And the cells that carry both variants of the plasmid, they are green and they are resistant. And for the monoploid chromosome, so these ancestral ones that are not resistant to kanamycin, I mean, they're resistant to tetracycline, but not to kanamycin. And the new ones, the new allele are resistant to kanamycin. So to compare our model then to these experiments, we needed to adapt it to their protocol, because what they do is like every 24 hours, they take a small sample of the cells and transfer it to new medium. And they do this for 30 days. And so we need to take this population dynamics into account in our model. And so basically what we do is we have this new allele, we start with it in a small fraction of cells, and then there's deterministic logistic growth. And then we do at this bottleneck, when they transfer, we make a random sampling of cells, and then we let them grow again. Okay. So then they were looking at the survival probability of this new allele in the population. So the survival probability is not exactly the establishment probability, because we are not starting with 1 copy in 1 cell, but with 1 copy in a small fraction of cells, but it's basically a very similar quantity. Now, what they did is that at every transfer, that's what you see like on the x-axis is the number of transfers, so basically time. At each transfer, they looked whether this new allele is still there or whether it has gone lost from the population. And they had 7 replicate population, and then they just looked all, they calculated always a percentage in how many of the 7 populations this novel allele was still present. That is survival probability. And that's shown here for a moderate initial frequency on top and a very low initial frequency of this new allele at the bottom. And we did the same in our simulations, just with many, many more replicates, not just with 7. And so that's our theoretical prediction here. And I think what I told you before about the establishment probability that this decreases with a plasmid copy number, that's also what you can now see here. So on the chromosome, which has a copy number of 1, the survival probability is much higher than on the plasmid. where like initially it's allele is present in all of the copies, but then here in this, for example, it goes to roughly 40% of the population that's retained permanently. And what's also clear is if the initial frequency is higher, the survival probability is higher. And I mean, those are then the experimental results that they got. And you can see, that our theory predicted quite well what's happening in the experiments. Okay, so it seems like our theory makes some sense. Let's go on. So all of this now was only looking at this establishment probabilities, where we saw that the establishment probability decreases with a plasmid copy number. But for the probability of evolutionary rescue, we also need this mutational input. And as I already said at the beginning, the mutational input increases linearly with plasmid copy number. Here with a plasmid copy number of 2, one would have 2 replication events at cell division. Here with 3, it's 3 replication events, and so on. And the question is like, okay, it's decreasing, the establishment probability is decreasing with a plasmid copy number, but maybe that can be outweighed by just having a higher mutational input. So maybe the probability of rescue actually increases. That's what we were wondering, which of the 2 factors dominates. So that's what I've shown before, just the establishment probability. As I said, the mutational input increases linearly in the plasmid copy number. And to get the probability of rescue, what one has to do is basically multiply sort of these 2 lines with each other. And if we do that, we see here for this dominant mutation that indeed the rescue probability increases with the plasmid copy number, but that's not the case for the recessive mutation, because here this drop in the establishment probability is so drastic that it cannot be outweighed by this increasing mutational input in the copy number. So now in all of this, I just said as a dominant mutation, there's a recessive mutation. What we did was a small model where we actually derived the dominance or recessiveness of this resistance allele from a small model of antibiotic degradation. So what's happening here in this model is that this gene on the plasmid is producing an enzyme, and the mutant version of it can degrade the antibiotic, rendering the cell more or less resistant, depending on like how many of this mutant copies it contains. And then, I mean, you can see here the growth rate of this sensitive population as a function of the external concentration. But through this antibiotic degradation that's in our model happening within the cell basically feels like if it has this mutant plasmid, it feels a lower antibiotic concentration. And that's shifting the curve. So for example, if here now 50% of the plasmids mutated, you can see that the growth rate is higher. If 100, then I mean, we get even more resistance. And now if we look at what's the probability of, what is the risk of resistance, so the probability of rescue depends on as a function of the antibiotic concentration. For different plasmid copy numbers, we actually see that this depends now, like which copy number leads to the highest probability of resistance depends on the antibiotic on the antibiotic concentration. So for example, this blue one, there's a copy number of 1, and you can, and for the red one, it's 10, so that's a 2 extreme. And you can see at this low antibiotic concentration, it's actually the red curve is higher. So a higher copy number leads to a higher risk of resistance. But as we go to this high antibiotic concentration, it's actually the low copy number that leads to the highest probability of resistance. Okay, so the optimal copy number from the perspective of bacteria depends on the concentration. And you can also understand this if you are looking at this graph again, asking what benefit does a cell actually have from having a certain fraction of mutated plasmids. And you can see here, if we are at this low antibiotic concentration, if just 50% of the plasmids are mutated, there's already a really strong fitness benefit. And having then the rest also mutated doesn't matter that much anymore. It's different here at the high concentrations, where like mutating 50%, I mean, it does help a bit, but it doesn't even get the growth rate above 0. But then there's a huge benefit of having also the second 50% mutated. We can then look at it in terms actually of the sort of dominance function, where we're looking at the growth rate as a function of the fraction of mutant plasmid in the cell. And you can see here for this low concentration, it's like a dominant mutation, not exactly, but approximately. For this high concentrations, it's more like a recessive mutation than the intermediate concentration it's between. So that's like dominant and recessive. And we've seen that for dominant mutation, the probability of resistance increases with a plasmid copy number. For recessive, it decreases. And that's then exactly in line with what we are seeing here. So here the high plasmid copy number gives the highest probability of resistance because the mutation is more or less dominant, while here where it's recessive, but it's a high concentration, it's best from the perspective of bacteria to have a sort of low plasmid copy number. Okay. So for all of that so far, we've assumed that actually the more of this mutant plasmid one has, the better for the cell, so the higher the fitness. But of course, there could also be situations where actually it's best to have both of them. I mean, it's a little bit unlikely an antibiotic resistance evolution. There are cases actually of antibiotic resistance genes where some alleles give resistance to one antibiotic and other alleles give resistance to different antibiotic. It's all like, I mean, this is then unlikely that we are having these two antibiotics during treatment of a patient, but of course it also could also be that bacteria are encountering both of them together in the environment, in which case it would actually be good to have both of these alleles in the population. So my student Ian looked exactly at this scenario where we are needing both of these different alleles to have rescue. And sort of this SMAX that you're seeing here, that's again, that's a maximum fitness that the cell or maximum growth rate that a cell can have. In that case, like if it carries both of these alleles. And actually now it's kind of clear that it's good for a cell if the copy number is high, because this allows for maintenance of both of these plasmid variants in the cell. You think of a single copy plasmid, that wouldn't be even possible. And so if you're looking here at the probability of evolutionary rescue, what one also sees, it's here shown as a function of the plasmid copy number. And if you just, so this is for different SMAX, but just focus on this orange line, you can see here that the probability of evolutionary rescue remains at 0 for this until we reach this sort of critical copy number. And that's exactly because, like, if you think of these heterozygous cells that carry both of these variants, there's always through this segregation process sort of this flux to this maladapted homozygous types. And if the copy number is too low, that's kind of this flux is so strong that it's just not possible for any of the cells to positively grow. And yeah, Ian also derived a condition for growth. So how much this as much actually needs to be such that rescue is possible at all, because you can see like here also with this orange, this critical number is different from the blue one, which has a higher S max. And so it's actually a very simple analytical condition here, also expressed in terms of the plasmid copy number. And yeah, Ian derived a super nice, he has a super nice mathematical proof also for this. Okay. So to sum up this second part, so we've seen that we have this multiple copies in a cell. What this means is that the establishment probability of mutations that appearing on the multi-copy plasmid, they are decreasing with this plasmid copy number. There's one caveat: if you're having gene dosage effects, then this can be different. But the rescue probability can either decrease or increase with the plasmid copy number. That depends on the dominance of this rescue or resistance mutation. Having multiple copies in the cell, this allows cells for to escape from fitness trade-off if sort of both of these alleles are needed. And also what we have seen in this comparison between theory and experiments, that a simple mathematical model can actually capture well this experimentally observed dynamics. And I should say that we really kept our model as simple as possible for that. Okay. So yeah, with this, I would like to thank my 3 PhD students, highlight them. They did the work that I presented today here are our experimental collaborators, especially Anna, who did the super cool experiments. This is my team. I also would like to thank Matt. So we have had many really nice discussions on evolutionary rescue together. Thanks again for inviting me here, and thank you for your attention.